I'm down in Florida now, and as you guessed it, I work on these guys, these little batty bats. And I wanted to step back and talk about why I do this, of course. I'm actually really interested in vocal complexity as well as social complexity. Looks like we're a little cut off. And of course, because of humans, of course I'm anthropocentric. We're all anthropocentric. But what I really am interested in is evolution and production. So I kind of have like a basic idea of where I'm coming from, and then we're going to talk about some singing and some bats and things like that. So, of course, we're here for sequences. But I'm actually really interested, there is the whole perception side, but what I'm really interested is in production and especially evolution. So, of course, we're going to have sexual selection and natural selection modifying, resulting in the evolution of these sequences, but that's going to happen through this production neurocircuitry, right? And then, yes, I'm missing some lines, I'm well aware, but we're going to try and stay simple. So one of my big goals here is that can we look at these sequences and go back the other direction? Can we look at sequences and can that tell us something about evolution, especially using the comparative method, and can it tell us something about the production neurocircuitry? And that's where I actually get to bats, okay? Why are they good for this? Well, there's over 1,100 species. They're all social, pretty much 90% of species are social. And they already have some pretty crazy production neurocircuitry for echolocation. They basically have a built-in auditory vocal feedback system. All right, so bats. Now we're going to jump a little bit, because that was kind of my big picture, and go to my title, songs. And the reason for that is because everywhere I go, I get, but do they really sing? But do they really sing? But do they really sing? And actually, so it made me think about it. What is a song? Okay, well, there's function, okay, and songs relative to calls. Bats also make tons and tons of calls. But if we think about it functionally, we're basically going to have a sexually selected signal here, right? Where it's going to be involved in courtship, territorial defense. But if we use that categorization, then all of these are singing. And in fact, you do hear about singing crickets, okay? You hear about, of course, songbirds, which is actually where I'm going. So this is a song, okay? But if I put a frog up here, that's a call, right? But this isn't a song. This is a call, right? These, of course, are songs, okay? And I don't have all these guys, don't worry. Now I'm going to add my guy. Don't mean to put him on top. All right, song or call? What is distinguishing these? So I argue that not only should it be about courtship and territory, and it doesn't actually even have to be the word song. We can come up with some other word. But there needs to be some difference when you end up with some sort of polysyllabic, multiple types of syllables put together in a sequence with temporal structure. Doesn't have to be a song. We can come up with another word. I'm okay with that. But for me, I don't like to pick up a paper and say it's a song and then look at a cricket call, okay, or a cricket vocalization. That's one of my big arguments. So why? Why am I like this? Of course, why does it fit here? Sequences. Well, why do I care so much? Well, it has a lot to do in part with grants, because our buddies here, the bird songs, are our model for speech. They are our model. And it's not because it's a sexually selected signal. Heck no. It's because it's a polysyllabic sequence of vocalizations, and because of the way it's produced. Specifically, bird song, besides the fact that birds fit in the lab a bit better than cetaceans, they really are the only accessible species that show these characteristics. All of these characteristics regular. Doesn't mean there might be some other species out there. Don't want to step on anyone's toes. But this is generally why you see birds as our model, and they are, and everyone will agree, they are our model of speech. They require audio feedback. They have syntactical organization. They have vocal flexibility. Depends on the species, how much, but you often have repertoires, and they're learned. Okay? So, what about my guy? Ha! You knew bats were coming in. All right. Require audio feedback. You guys already got kind of the preview on that. I haven't even done it with song, but that's because if you deafen a bat, it dies pretty much. They can't even see if you deafen a bat. They just can't handle it. They can't eat, they can't function. So when a bat is flying, this is an example of a bat echolocating. Notice they're producing this sound based on the incoming echo. they're going to modify their outgoing signal, and you have basically intense auditory feedback, audio-vocal feedback going all the way through all the time. So, yes. Okay. Syntactical organization. And I'm going to actually put this in as well. Okay. So, what do these guys look like? I know. So, this is actually a captive colony, and this is a territory, and these are a bunch of girls in here, and this is a bat, and he's going to do a wing flapping display and do his little song. I have slowed things down, although you can hear them, and you'll hear them later. Okay, so that's kind of an example. So you get a little bit of behavior with it. What are they doing? So how are they putting, let's go straight to how they're putting these things together. I'll just play a couple of them. Let me skip, this guy I can play so you'll get, it kind of helps to hear these things. And notice I did slow them down more than you can hear them actually at halftime, but it makes it a little easier to follow. Kind of getting the idea, right? Now, I picked these songs. You'll see later. I picked these songs because they show this kind of. All right, so if we put these together, what do we have? We have three types of phrases, right? Chirp phrases. You can tell in these chirp phrases, even though each of these males are different, that they're really built from the same syllables. Type A and type B syllables. And you're probably wondering about these guys here. Yes, they're extremely, extremely stereotyped. And if you look at the bats, this is from 50 different recordings of each bat. And some of these guys some bats I've recorded over a 4-year period. Their type B syllable is their type B syllable. So we have our chirp phrase. We have what I call trills. They're these short guys here. And then we have buzzes. They're not quite as exciting, so I haven't zoomed in on them. And so how are they putting these songs together? So if we look at sequences, what I actually look at instead of syllable sequences is I look at phrase sequences. So for example, this top guy is chirp, buzz, trill, 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 buzz. Second one, chirp, trill, trill, buzz, etc. So you're probably thinking all the songs go, somehow from chirp to trill to buzz, but keep it in mind, I've picked these guys. Now, one thing, though, that happened is when I look at every single phrase, I get so many different variants. It's kind of hard for me to make heads or tails. So what I did is I took out the repeats, right? So instead of calling it chirp, buzz, trill, 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 buzz, I just called it a chirp, buzz, trill, buzz song type, right? And I have kind of song variants and song type. So if we look at these guys, we got a chirp, trill, buzz and a chirp, trill, buzz. So we're kind of getting the idea. So you're thinking, wow, so do the bats produce the same song every time? The answer is, heck no, right? They actually produce a lot of variants. So you can tell that this is the same bat, but this is a chirp, trill, buzz song, and this is a chirp, trill, chirp song from the same bat. Now we go to a different bat, whoa, totally different type B syllable, and we have yet another song type, a chirp, buzz. So how many song types? Well, this was when I had, I guess up to 60 songs. I've gotten up to about, for song types, and this does not include repeats, I seem to be asymptoting out at about 60, and I haven't hit an asymptote when you include repetitions yet. Just kind of keeps going, okay? However, it's definitely not random. You have a lot of song types that are shared. That is, notice 55% of bats produce chirp, trill, buzz, which is why those were the ones I put up there, because they have all types of phrases, and it's a common type that's produced by the majority of bats. Okay, so putting these together, I did a very simple Markov model to try to figure out what rules, can we come up with any rules for these? And I have to say, and we're now looking at that there's better models to look at them, but we were able to come up with some basic grammatical rules. That is, you generally start with a chirp, buzzes usually happen towards the end, and you never go from a buzz to a trill. Okay, so at least we've got some sort of syntactical rules. By syntax, of course, I mean order rules. There's no referential here at all. So, where are we? Echo, yes. Syntactical organization, yes. Vocal flexibility, yes. All right, but let me go for a quick, so you're probably now all wondering, why do they have all these song types? Right, so I'm going to go for a very rapid side tangent, because if I don't, everyone's going to harass me all the time and wonder why they have all these song types. So I'm going to do this really fast. Okay, so a little bit about these bats. These are the most abundant bat in North America. And this is, I was first started working on these guys in Texas. Their populations, this is kind of a continuous migrating population. However, they also live in Florida. I'm recording them there. I've recorded them all the way down into Mexico, Honduras. They go all the way down to Brazil. Okay, the species. Now, the population I started all this on was here in Texas. Basically, the females come down into Mexico during the winter. They come up north, and they form these huge maternity colonies that you hear about. So it's a wonderful little side note. You go into look at the Doppler, and it looks like it's raining every single night in Austin and San Antonio, or near San Antonio. No, those are all bats coming out. Those are all females. So here I am in Austin. We get all these females coming in. I'm working with a bat rehabilitator, and we only have 5 or 6 males compared to all females. And so he said, so where are the females? And they're like, oh, where are the males? The males are down in Mexico. They're definitely mating down in Mexico. So I go down to Mexico, and I'm going, so where are the males? They're like, oh no, they're mating up in Texas. So we're looking everywhere for these mating males, and I happened to talk to Mike Smotherman over at Texas A&M, and they're mating at one of the big, huge male populations is at the Texas A&M football stadium. So in fact, all these lines, so they all live inside these cracks. This is all guano dripping down. And basically what happens is the females, they head south, they come up, and they come up through College Station in the spring or late winter. And the males basically hang out year-round, waiting for the ladies. The ladies come through, and it's one giant orgy, basically, which I think is absolutely hysterical, because this is Rick Perry's alma mater. And it is the largest mammalian orgy, I think, in the world. Okay? So what this made, though, so what was wonderful is I had all these bats. These guys also, so we could basically go out in the field, right? Underneath the bathroom and the concession stands, right? But notice where these bats are. Trying to figure out what the heck they're responding to is impossible. But luckily, these guys are really mellow bats. That's another whole story. They take very well to captivity. So what I'm going to show is what you started to get an idea that these guys, the cues, one of the main cues of these guys, shocking, I don't know why it didn't hit us, are auditory. Okay? And so what we noticed, so this is basically what they do is they set up territories just like they did in another captive colony. We have these big tubes. There's a male in here. He's got 3 females. He sings. And what I've done is this is my ultrasonic mic, but what I'm actually going to play for you is my regular mic. Note, you cannot hear any echolocation on my regular mic. However, you can hear their songs. So every vocalization you hear is actually a song. And wait till you see what it's correlated with. Oop. I can get my... Oh, that's kind of a problem. Try from there. Those are their little feet up against the... Oh. So, watch a little more. And... and then we started noticing every time the bat flew by. All right, so we thought, it might be echolocation. And there's a lot more to the story. But what we then did is we looked at these. These were the percentage of song types and during echolocation experiment playing backs compared with spontaneous songs produced in the roost. And that was the best we could get at social context. We did these experiments during the day. We did playback experiments using echolocation. And first of all, you play back an echolocation call, I can go pretty much, I've now done it in Florida, I can go anywhere. You play an echolocation pass, you get song. And you get it within about 300, 400 milliseconds. So that's a very big stimulus. And so what I did is I compared what we got in response to echolocation call was spontaneous songs. And we performed these experiments during the day. So it was actually remarkable we saw something because spontaneous could include just about anything except for echolocation. Because echolocation, we were doing it during the day and there were no bats flying. But God only knows what all was going on in that roost. It was up in that crack. But we still see basically shifts in the song types based on the social context. So it looks like, we haven't gotten to female preference yet, but it looks like it's not going to be some sort of, like a one-to-one correlation, but song type does seem to be affected by social context. Okay, so the learned. We're going back. That was my quick side tangent. I'm not sure how I'm doing on time. I started a little late. Wait a minute. I started totally late. I'm not out of time yet. Ah, yeah. Okay, so why do I care about all this? Well, remember my original picture, vocal production, blah, blah, blah. That's why. So you've got our human speech. I'm going to actually get all these up so I can go fast. All right. Why do we care? Why do we care about the call circuit, the calls and the songs? Because they require or appear to use different circuitry. And the whole point of songs is that this circuitry, and I'm not going to go through all the neurobiology, but I think you guys can see the parallels between the birdsong circuitry and the human speech circuit. One of the biggest things is this basal ganglia thalamic loop, which is analogous to the area X DLM loop. It is this loop that produces sequences, and it is this loop that's involved in learning. So you have some sort of circuit that's going to allow you to incorporate an individual syllable, note, whatever, call into a sequence. That's going to require different circuitry than just producing one set of vocalizations over and over again. And in fact, all mammals, other than humans, thus far, they've not been able to find anything but this. Okay? Doesn't mean it doesn't exist, but they haven't been able to find it yet. All right, so what about my guys? Ha, you dying? Dying? All right, well, what happens when they echolocate? So if you look at CFOS, comparing bats that are echolocating and non-echolocating, basically look at what parts of the brain are active. Of course, we get the ACC and the PAG, but we also get the basal ganglia, so which you normally do not see in vocalizations of any other mammal. Doesn't mean mammals don't use a lot of the basal ganglia. And the basal ganglia is also involved in motor control, makes sense, learning, motor sequences. Okay, so for the neurobiologists out there, this is with Mike Smotherman, just kind of showing the parts of the basal ganglia that are indeed involved in echolocation. All right, so what happens when you stick, basically pharmacologically, add some drugs that activate those parts of the brain? Okay, it sounds really evil, but you would be really shocked. These bats are so hardy. I didn't do the surgeries. I'm not very good at that stuff. But Mike would put these things in. We'd take them out, and that bat would live in our kennel, happy as could be for a long time. It was these are actually very robust bats. It was also nice and refreshing to know that we had about 1 million of them living 2 blocks away. So, but when we do this, what happens? When we turn on the basal ganglia and thalamic loop area and the ACC, what do we get? Well, we get vocal sequences. So, not just echolocation. In fact, at very low levels, we would get echolocation. As soon as you add higher levels of activity, we would get no echolocation. All we would get these are obviously social communication calls, sequences, over and over and over and over again. And what's even more remarkable is we actually were getting song-like sequences. Okay, and so, it's not quite right, but notice we do have a chirp, a trill, and a buzz, just like we kind of did with our original. And this is not the same bat, by the way. This is just kind of giving an example of the different types of phrases. Okay, so what was actually, this is not quite as important, but we could actually go from not song, no song syllables at all, to pseudo-song, right, kind of at least part of it, to song based on which drug we were using. So we were definitely affecting specifically the production of sequences, song sequences. So what are we looking at now for a model with the free-tailed bat? Something like this, right? So we're pretty excited about it because we're getting something with sequences, and this also relates to why sequences are important. So remember, though, this is kind of where we started, right? And especially my model of production and complexity. So when we again think of sequence models, and remember, you didn't think they were social. You walk into a colony, and everyone, I'm sure, is trying to record like one bat out of here is a nightmare, because these are not quiet bats either. So I'm only talking about song today. They have all sorts of calls produced in other contexts as well. There are sequences. They do all. These guys are crazy little bats. But there are actually over 150 species from this family. And so far I've recorded 5 different species, all producing vocal sequences. So one question, of course, is the neurobiology of vocal production, like as I've kind of started out with as being an interest of mine. But I'm especially interested in the evolution and being able to do interspecific comparisons. What do I use as a metric? Okay, that is, especially if we want to think of a complexity metric. But for me, a complexity metric is going to relate to the neurobiology of vocal production, the complexity of the circuit that is required to produce the vocalization. So that's kind of where I'm coming from, and that's it. Yeah. Yes, so they, both of them hit the dopaminergic system. Canic acid, though, is a lot stronger than bicuculin. So basically with the canic acid, we would lose, it was almost like we would just fry out the circuit, if anything. They would just lose, they would produce these sequences, but they were basically producing what I would call gobbledygook sequences. I can talk about it more, but yeah, I do have all the info on that.